Good morning, or good afternoon, as the case may be. Hey guys, and welcome back to another great week. I hope great anyway, or to another week, however you feel about it, of quarantine. We're going to go ahead and this week we're going to take a look further into what we've been learning about in the cell cycle and the inheritance of genes and all that cool chromosomal stuff. And that's really all we're going to do this week is really look into that, make sure we really understand it before we move on to the next thing. So if you guys remember, last time we were talking about meiosis, and we said that meiosis was the process of sexual reproduction and division. So we would have one person, right, who we'll go ahead and say this person is male. And this person, who looks fantastic, by the way, amazing artistry here by Mr. Garner. So there's the male, and then over here is another person who's a little bit bigger. It seems they're giants, they're huge. Um, this is the let's say female, which now I feel bad about calling them a giant. They're not a giant. They're normally proportioned. He's standing further away from the camera. Anyway. So we have the male and the female, and they undergo meiosis, and undergoing meiosis in their specialized precursor cells, remember their germline, those cells will undergo meiosis, and they're going to create their specialized sex cells called gametes. So they're going to undergo meiosis, which is that 8-step process, and here we have their haploid gametes. And remember we said that haploid just means that they have half of the chromosome number, or they have one of each chromosome. And so over here with the male, this is going to be the sperm cell, right? And over here with the female, that's going to be the egg cell, right? And so these two are going to be added together, right, eventually during reproduction, and they are going to create the zygote, which is the first new diploid body cell of the developing embryo. So this one over here is going to be the diploid zygote. Okay, and so when the diploid zygote is created, that diploid zygote is going to get the same number of chromosomes from dad and from mom. So it's going to have 2 of each chromosome, that's why it's diploid again. But at each chromosome pair, such as chromosome pair number 1, because we used that a lot as our example before, they'll have 1 chromosome at that pair from dad and 1 chromosome at that pair from mom. They'll have the same genes, and these are known as homologous chromosomes. They'll have the same genes in the same order, in other words, on that same order on that chromosome there, but different alleles. And remember, alleles are slightly different versions of the genes. And so they'll have mom alleles on one and dad's alleles on the other. And those together will produce the proteins in that zygote's body that will make up its characteristics. And so since it's a mix of the 2 parents genetically, it ends up being a mix of the 2 parents phenotypically as well. Okay, but so why do certain children end up looking like one parent more so than the other? Well, the reason for that is that certain genes are dominant or recessive. So if you remember back, we can have a recessive, which I'll just go ahead and type this. We can have a recessive allele, which means that it creates the recessive phenotype or appearance only when the dominant allele is not present. Okay, so in other words, they haven't gotten that dominant allele from either dad or mom. And then the dominant allele creates the dominant phenotype when, now I guess I could say only when, at least one copy, this thing's really getting in the way, isn't it? Oh, sorry about that. At least one copy of the dominant allele is present. And so we, to go ahead and to clarify that, when we were taking a look at that chromosome pair, number 1, I said that we could have the condition where our dad might have the dominant allele and our mom might have the dominant allele. And remember, dominant alleles are shown by capital letters. And so in this case right here, big A would equal the homozygous dominant condition, and this person would have the dominant trait or phenotype. Um, or that person could inherit, let's make the mom dominant this time, that person could inherit a recessive allele from dad, and in this case, when we looked at their traits, they would have a big A and a little a, a dominant and a recessive, and this would mean they were a heterozygote or a hybrid, and that would mean that they would still have the dominant trait because they have a copy of the dominant allele. Only one copy is needed. Or we could have the condition where both parents have the recessive allele, and in that case, we're going to get that recessive condition, which is homozygous recessive, and that means that they're going to show the recessive trait or phenotype. Right, and so this is why certain organisms are going to appear to favor one parent over the other. They got a dominant allele from that parent or recessive or whatever, and they're showing it based off the combination. Now, for each one of your traits, you have at least 2 alleles that are coding for it, but there are many traits that actually have many more alleles coding for them. And we'll talk about that in a little bit. Those are polygenic traits, right, which makes sense. Many genes, of course, are the things in your DNA that are coding for your proteins. Many genes for one trait. But here we're just going to assume, and for most of our practice problems, we're going to assume that there are only 2 alleles for each, for that particular trait. Okay, so hopefully all of that makes sense, and that's a good recap of what we've gone over. Today we're going to go ahead and apply that. So we said, we talked a little bit last time about Mendelian inheritance. Essentially, Mendelian inheritance is describing how certain traits are inherited based on the alleles that you have. And so Mendel was that wonderful Austrian monk who was like, haha, I will do the experiment with pea plants. You can't stop me, even though he was turned down for the mice. And he came up with that brilliant way of showing inheritance in the Mendelian ratio. So what he came up with was the golden ratio, of the monohybrid, cross, which was 3-to-1 dominant to recessive, right? And so if you remember, what he did was he took a bunch of pure breeding pea plants, which means pea plants that had the particular traits he knew he wanted, and so he figured the particular genes he wanted. And so he went ahead and he crossed those in something called a monohybrid cross. All right, hang on just one second. Okay, so he didn't start off with a monohybrid cross. What he actually did, let me grab this here really quick. The whole thing, please. What he did was he started with one cross where he took 2 pure breeding plants. I'm just going to say big A, and little a. So he took the pure breeding plants for recessive and dominant, and he went ahead and he crossed them together. So he had his big A, and then his little a. And all of the organisms that came out of that cross were big A, little a. They were all hybrids. They were all heterozygotes. And they all had the dominant phenotype. And so that led him to conclude about that dominance thing. He was like, there must be something about the traits of one organism that can override or overrule the other. And in this case, he saw that was true. They're 100% dominant phenotype. He then took 2 of these organisms from that cross, and he crossed them together. And he went ahead and made his little Punnett square here. Okay. And he went ahead and he performed that cross. And what did he find? Well, he found that golden ratio for the monohybrid cross. So here we're going to see it made over here big A, little a, little a. Move that over slightly. And little a. And so what we see here happening is these 3 are going to have the dominant phenotype. Let's say that's purple. They're going to have the dominant phenotype because they have at least one copy of that dominant allele. And then these guys over here, let's say green is the recessive, these guys are going to have the recessive phenotype because they are going to have 2 small a's and therefore have the recessive phenotype. So over here we have 75% is dominant and 25% is recessive. And if you're wondering how I got those percentages, Well, if each one of these, if all four of these boxes equals 100% of their children, each box is 25%, right? Because 100 divided by 4 is 25. And so this is 25, 25, 25, 25. And so I added up these three to get 75, and then the last one, 25, is recessive. And of course, remember, these are just predictions. These are not hard and fast. This isn't going to be the case for every parent that has four children. They're going to get three that are the dominant phenotype and one recessive, but this is statistically a way of measuring it. Right, so this is what we talked about with the golden ratio of the monohybrid cross. In other words, we're looking at one trait, which is the mono, and hybrid over here. This is a true monohybrid cross. I'll draw an arrow to it. This one over here. Here we can see, because we're crossing two hybrids, they are both hybrids for that one trait, and this is the ratio that we get. So that was a very important ratio, and basically what it showed was, yeah, there's a pattern to how the stuff is inherited, and therefore there has to be some sort of function as to, or mechanism as to how it works. Okay, so Mendel knew that all animals and organisms had far more than just one trait that he could look at. He was, over here in monohybrid traits, he was looking at flower color, or he was looking at seed pod color, or he was looking at the height of the plant. He was only looking at each one of those isolated from all the others. But Mendel knew that that wasn't, entirely correct. I mean, there are many, everybody has many different traits. You are not just eye color. You also have, hair color and skin color, and you have different heights and different shapes and all that stuff. And so was it possible, he wondered, to be able to, we already kind of covered this, was it possible, he wondered, to be able to look at several traits at once? And the answer is yes. So he came up with something called the dihybrid cross. Okay, so the dihybrid cross, let's take a look at that. So Mendel, he went ahead and he crossed two different pure breeding lines of plants, and he had actually crossed them to be pure breeding for more than one trait. And so he had determined, he had taken pure breeding tall purple flowered pea plants, and he had crossed them with pure breeding, or homozygous if you want to say, short white flowered pea plants. So he went ahead and he crossed these two. So you can see we're looking at two different traits here, right? We have the trait of height, which I'll show here, so tall versus short, and we have the trait of flower color, purple versus white, right? So we have two different traits here that we're looking at. And so he had just he went ahead and did the cross, and all of the offspring came up as tall and purple flowered. So he knew that tall and purple were both dominant traits. So I'm going to put that over here. So typically what you'll do when you're determining the letters for this is you'll take the letter for the allele will be from the description. So if tall is the dominant, we would say capital T for tall is the dominant allele. And we'll say that equals tall. Then you would take the lowercase to denote the recessive. Okay, so little t would be the recessive, and that would equal short. I know short does not start with t, but it's always based off the dominant. So we have t, big T equals dominant, which is tall, little t, which is recessive and short. And then we're going to do the same for flower color. So big P is going to equal our dominant, and that is going to be purple flowers, and little p is going to equal our recessive, and that is going to equal white flowers. Now, can you do it so that you use a P for purple and a W for white? You can, but I will tell you, for reasons that you might see if you try it, it'll become very confusing. So it's best to use the same letter, just different cases. If you find that the P's are too close to each other to be properly told apart from each other when you're doing it, then you can use any letter you want to. So sometimes, if we were saying that the trait was silver flowers, I wouldn't want to use an S because the capital S looks very much like a lowercase s. I could use whatever letter I really wanted to do that. So we go ahead and we set this out over here. This is always a good guide to set off to the side. And he went ahead and he did this cross. So let's look at that original cross there, okay? So that original cross, here's our pure breeding tall purple flowered pea plants. How do we look at that? So pure breeding tall is double dominant, or homozygous dominant for tall. And then pure breeding for the purple flower is homozygous dominant for the pea. So it would look like that. And then the same for our short white flowered plants. So this is his original cross right here. So this one right here that I'm talking about, this is that cross. And so how do you actually draw this out for your Punnett square? Okay. So I'm actually going to draw a box using this thing because I don't think I could do it just freehand. So wow, and that box got really, really big, much bigger than I wanted that box to be. Wow, that box just keeps on going. So I need that box to be a different shape. Thank you, computer. Okay, so something like this. So you're going to draw a box, and then you're going to go ahead, and I'm probably just going to want to draw lines in as well. So you're going to do 3 lines down, 3 lines down, and these should be equidistant from each other. I just obviously suck at drawing for this one over here. Go away. There we go. So you're going to want 3 lines down, and then you're going to want 3 lines across. So 1, 2, and 3. Look at that. That's not bad. Okay, so this one could be moved up just slightly if I could. Yep, there we go. Awesome. Hey, that wasn't bad at all. So this is what you should end up with. Now, so you're probably wondering how many boxes does that give you? Well, you have 4 down and 4 across. It gives you 16 boxes. Why 16? Well, because when we were looking at the, each trait has 2 alleles, right, that codes for it. And when we were looking at the monohybrid cross, that meant that we had 2 for each, even though we were looking at just one trait, it meant that we had, each parent had 2, and therefore we had 4 altogether, so 4 boxes. Well, now each parent is 4, so 4 times 4 equals 16. That's where you get those 16 boxes. Okay, and the other difference between this one, I'm going to get rid of this guy because I don't need it. The other difference between the monohybrid and the dihybrid is see that we have 1 letter per column and row here. Over here, we're going to have 2. And in order to explain this, I'm actually going to go and talk about one quick thing before I go into this. Okay, so remember that here, all of this big A, little a, big A, little a, this all came out of one parent having big A, little a, and then the other parent having big A, little a. So what you must never forget is that this all is referring to these organisms' chromosomes. So we have the big A, and then we have the little a, right? And then we have the big A, and we have the little a. That's what we're really talking about, is that these alleles are on their chromosomes. So when we talk about it with the dihybrid cross, all right, how are we going to do this? Because we're working with a lot more traits. All right, so I want to break something down really quick for you. All right, so remember back to meiosis, we made gametes, right? And so at the end of meiosis, which I'll draw over here, we had our first cell, right, which was our precursor cell. We ended up getting the first division, right? And then without renewing our DNA, we went ahead and did our second division of meiosis. And it gave us 4 haploid gametes, which I'm going to try to draw with any kind of grace here, and it's just going to fail. Okay. So we have 4 haploid genetically distinct gametes, right? Okay, each one of those gametes, if you remember, based off of the laws of independent assortment, oh my God, I can't think of the other one right now, but you know, the law of independent assortment and the other one that would shuffle them, based off of those, each one of these got a slightly different allele and chromosome, and so when it was combined with another gamete or another sex cell, it would end up coming up with a unique, oh my God, what's wrong with me? With a unique combination, right? And technically, and I wonder if I could go ahead and, yeah, I think I can. I'm going to actually copy this guy over. Oops, don't want that. Sorry. Okay, so technically, when we're looking at this, and let's ignore for now that whole polar body thing with the female's gametes. We know that they don't make 4 identical gametes, they only make 1. But ignoring that for now, okay, we can have any potential combination of mom and dad's gametes, right? So we could have this egg cell combined with this sperm cell, or we could have that egg cell combine, or we could have that egg cell combine with this sperm cell, right? Or we could have that egg cell combine with this sperm cell or this one, right? Or we could have this egg cell combine with whatever sperm cell. So you kind of get where I'm going here, hopefully, is that there is an infinite, well, not infinite, but there are many ways that we could combine these 4 different chromosomes with this one. In fact, if we were to look at it statistically, what we would figure out is since there are 4 different combinations over here and 4 different on the right, it would be 4 times 4, which would equal 16 different possible combinations that we could get in terms of our gametes. And that's funny because we have 16 boxes. So, aha, it's all coming together just as planned. So anyway, so that means we end up with 16 boxes. Now, how do we actually determine what's going to go over each of these boxes? Well, I'll tell you. So what we're first going to do is we're going to take a look at that one parent's genotype, big T, big T, big P, big P. So this is our first parent, the purple tall flower pea plant. Okay, so if we take a look at this organism in its genotype, there are different combinations of genes that we could get using this guy's allele. So because these are two different traits we're looking at, we're going to treat them as if they're on different chromosomes. So the T trait is on a different chromosome than the P trait. And that's going to allow us to do the following. We're going to try to come up with all the combinations of alleles that this organism could produce. What do I mean by that? Okay, so the first combination I can see is we could take this big T and pair it with this big P. And right now, if you're wondering what I'm doing and you're like, I'm totally lost, hang on for just 1 second, okay? I promise it'll make sense in a second. Okay, so that could be the first combination I could have, big T, big P. Okay, I could then see combining this allele, this chromatid, with this chromatid over here in this sex cell, and getting this combination, TP. Looks the same, but I know. Just hang in there with me. Okay, or I can imagine taking this T and mixing it with this P over here, and that would give me the combination. Okay, or I can imagine taking this T and this P over here and combining them together, and that would give me this combination. So these are all the possible combinations of alleles that I could get using that person's genotype. So in other words, these are all the different ways they could segregate during the laws of independent assortment and the law of segregation, I think is the other one, which puts them together into those gametes that could then combine with somebody else's. So these are all of that person's potentials. I'm going to write those up here above each one of the boxes. So here's the first potential, the second potential, the third potential, and the fourth potential. So these are all the potential combinations we could get on those gametes or alleles. Now, what are all the potential combinations we could get with the other one? Well, the other one looks like this, and you can probably imagine that these are all the combinations we could get from that one is just big T, or excuse me, little t, little p, little t, little p, right? Because we would do the same thing, and that would give us this. You're probably wondering, I don't get the point. I don't understand what's happening. What's going on? Basically, if you remember back to meiosis, that random shuffling of chromatids is going to determine what you get as a child. So whether you get dominant or recessive or whatever. And so what we're doing here is we're pretending to do that. We're pretending to shuffle these up to get all the possible combinations that we could get when these two individuals reproduce together. And so then what do you do? Well, you go ahead and you just distribute in the boxes, big T, big P, little p. I'm just combining these two guys right here. I'm doing the same thing over here. And what you'll notice is that all of them are actually going to be the exact same. So I'm not even going to fill up all the rows. I'm just going to say that they are all going to look like this no matter what. Whoops. Sorry about that. Mixing my T's and my P's. It's not good. Okay. So these are all, this is the one possible combination we could get from these pure breeding. So when Mendel bred these pure breeding tall plants, tall purple plants with the pure breeding short white plants, basically what Mendel was getting, the reason why all the offspring were exactly the same and why they were all tall and purple is because they were all inheriting at least one copy of the dominant allele for each of those traits, for tall and for purple. And so that's what he was ending up getting, was something that looked like this, right? And all the plants were tall and purple. Now, he took two of these and he combined them together, right? So he went ahead and he did the big T, little p. He took, each two of these organisms, these offspring, and he went ahead and he combined them together into what we call a proper dihybrid cross, because they are two hybrids and they are hybrid for both traits, right? Hybrid for two traits, dihybrid. Hopefully that makes sense. Okay, and so then they went ahead and did this. Now, what's interesting about this is that when we consider all the possible combinations here, we get something a little bit more exciting than we did before. So if we imagine we do the same thing that we did last time, so I'm going to try to make it the same color. I did blue, red, purple, green, okay? So the same thing as last time. I distribute this allele to this one over here, making this possible combination. So big T, big P. Okay, and then I go ahead and I distribute that allele over here to this one. So big T, little p. And then I do this one over here to this one, and that's going to be little t, big P. And then this one over here to this one, and that gives me little t, little p. So these are now all the possible combinations or shuffles of alleles that we could get in our gametes in our sex cells, and that's going to produce a much more exciting dihybrid cross. And so if we were to go ahead and run that dihybrid cross, which I'm not going to do here because hopefully you guys have figured it out over here, but it would be the same thing. I'll go ahead and I'll just, I'll go ahead and I'll just change these. So that one can stay. The next one was big T, little p. The next one was little t, big P, and then both of these became small. So that's what we would get on that side. And then because the other one was the same, they were both perfect hybrids. This is what we would get on the other side as well. Okay. For some reason, I put the there we go. Okay, and so we'd end up with something like that. And so now if we were to go ahead and start putting them in there, we would find that they would change actually quite a bit, that each one of these would be very different. And so we would need to do all of the different boxes. And so it would look something like this. And obviously, I need to get rid of those lines because no, not all of them are going to be the same anymore. I'm sure there's a way to get rid of these in a better way. Yeah, okay. Cool, anyway, so good. So anyway, so we keep filling out this box, and I am, in the interest of time, I might not fill it out all the way, but you can continue to fill it out. I'm literally just distributing the T's and the P's. And hopefully correctly. I'm sort of doing it as fast as I can here. Hang on. You can just speed up the video to the next little, when I'm done with this, if you guys can see it. I apologize for this. This is new to me and figuring out how to actually teach this stuff this way. So almost done. Boop-a-doo, ba-doop, ba-doo. I hope that was right. Okay, yeah, this looks right. Cool. So then it was something that looks like this. All right, so Mendel got this. And what's interesting about this is that there are many different genotypes that we could get now. And as you can imagine, the phenotypes were quite different as well. But the phenotypes, there weren't as many different phenotypes as there were different genotypes. Why? Well, because that dominant and recessive rule still plays out. As long as you have one copy of the dominant, you're going to show that dominant trait. So we're actually going to have more genotypes than we will phenotypes. The phenotypes will some of them will be overridden by the dominant. Okay, because we really only have two phenotypes for each one, tall or short for the first one, or purple and white for the second. So if we were to go ahead and write out all the different possible phenotypes, we could have tall and purple. We could have short and purple. We could have tall and white, and short and white. And those are the only four phenotypes that we could actually have out of this cross, which is really wild to think about. But those four phenotypes are really all we have. And so if we take a look at that then, we're going to end up, and we were to go ahead and count how many of each one we had. Let's go ahead and do that. Over here, we have, yes, that would be tall and purple. Yes, that would be tall and purple because they have at least one capital letter for each one of the traits. Yes, yes. Yes, no for this one. This one would be tall, but white flowers, so we're not going to count it because there's two little p's. Yes. Same over here, tall but white. Yes, yes. No, this would be short with purple flowers. We don't want that. Short with purple. Yes. Good. And so that's all of them. So let's count them. 1, 2, 3, 4, 5, 6, 7, 8, 9. So we're going to have 9 tall in purple. Now let's take a look at short in purple. And you can pause the video to see if you could figure it out before I do. Okay, I'm assuming you pause it. So let's look at the short in purple. So we want 2 little t's and at least 1 big P. So there we go. There's 1, 2, and 3. Okay, so we have 3 short in purple. Now we're going to look for tall in white. All right, 1, 2, 3. Okay, and now we're going to look for short and white, which is easy because there it is right there, just 1. So this comes out every time, Mendel found, with a 9 to 3 to 3 to 1 ratio. And this is what he called the golden ratio for dihybrid crosses. This was very exciting to Mendel because it proved that there was an underlying pattern to inheritance and showed that, yes, there was some logic as to how this stuff was being passed on. And that meant if you could predict what was being passed on, you could do a lot to figure out what was the mechanism that was actually driving that. Of course, we know it's genes and alleles, but he didn't at the time. And so now we know it's dominant and recessive alleles that are being inherited in a certain pattern that are allowing this ratio to be true. Okay, so I didn't expect that to go on for quite that long. I guess we'll go ahead and stop here because I don't want to keep going. It would be an incredibly long video if I did. So what we'll do is we'll do some dihybrid practice and stuff for this particular lesson. And then what I'm going to end up doing is on Wednesday, we'll go ahead and talk a little bit about actually, I'm gonna on Wednesday we'll talk a little bit about inheritance, and a little bit more on this, and then on Friday we'll just go ahead and take a quiz on what we've learned so far. I don't want to go any further, and then next week we'll go ahead and start applying some of this stuff. Again, if you guys have any questions, anything for me at all, please do not hesitate to reach out to me via email. Or the Google Classroom, or the Google Voice number if I've called you guys, and I'll try to put that up on the Google Classroom soon. I keep forgetting to do it. I hope you guys are staying safe and well and enjoying life with the stay-at-home order. I think we're all gonna be going crazy at home soon, but hopefully this will keep you occupied. All right, guys, take care.